So today we will start our next unit, that is the gene regulation in prokaryotes. So why there is a need to regulate a gene? Now in order to exist in the environmental conditions, different environmental conditions, every organism has to regulate some genes in order to survive in these conditions. So to that purpose, the gene regulation is playing a very important role in every living organism. And these gene regulations, they helps this organism to get adjusted to the different environmental conditions like high temperature, low temperature, high pH, low pH. So according to that, these bacteria, these prokaryotes, they can regulate their genes in order to survive in these conditions. So these gene regulations, they are playing a very important role in prokaryotes in order to survive the different environmental conditions. So what is gene regulation? Gene regulation, it is nothing but a mechanism which collectively controls the product of that gene or synthesis of that product of that gene. You can simply define this gene regulation as the synthesis of a particular gene product is controlled by a mechanism which is collectively called as a gene regulation. Means if a protein has to be produced, that protein must be produced at particular time, and that time is decided by this gene regulation mechanism. Now these microorganisms, they are able to control their gene expression which is present in their genome. For example, in E. coli chromosome, it contains large amount of information, large amount of nucleotide sequences which contains the information needed for that E. coli cell, and on that basis which information is present in the E. coli chromosome, that E. coli chromosome contains such information from which near about 450 polypeptides can be produced at a time. Such large amount of information is present on the E. coli chromosome. But all these 4,500 polypeptides, they are not produced at a time. They are expressed or they are produced according to the conditions which this E. coli bacteria faces. So this is the regulation. Means all the genes, they are not expressed at the same time. And this expression is controlled by a collective mechanism. That mechanism we call gene regulation. And through these gene regulations, this microorganism expresses only those genes which are required at that particular condition. So what this gene regulation helps these organisms? This gene regulation helps this organism to conserve energy and raw material which is needed for that organism to survive. It also helps this microorganism to maintain the balance between the amounts of various proteins. At a time, some proteins are needed. So in order to produce the proteins, large amount of amino acid will be needed. If suppose that organism is an autotrophic organism, means it can synthesize its own products. At that time, if a protein has to be produced, amino acid must be made by that bacteria in order to produce that protein. So to avoid such hard conditions, these bacteria, they can regulate their genes according to conditions. So because of that, they are able to maintain the balance between the amount of various cell proteins. And this gene regulation also helps these microorganisms to adapt to long-term environmental changes. So whatever the environmental changes that happens there, according to that, these microorganisms can change their gene expression through the mechanism that is gene regulation mechanism. Now, in microorganisms, there are some proteins or the enzymes, they are continuously needed for the microorganisms. You can say that, for example, those enzymes which are involved in energy producing mechanism, as you know that the EMP pathway, then TCA cycle, then energy building, that is electronic transfer pathway. These are the pathways which are continuously run in every microorganisms. So to run these pathways, they require this microorganism requires certain enzymes which are constantly produced there. And these enzymes which are constantly produced in these organisms and their function is constantly needed there. So such enzymes which are performing this important role in central metabolic pathways and their functions, the functions of these enzymes, they are known as housekeeping functions. Means these enzymes, they performing very important function in order to maintain the balance of energy which is required by that cell. If EMP pathway is not there, then no ATP will be generated. TC pathway is not there, no reducing power and energy is developed there. So these enzymes, they are constantly functioning in these bacterial cells and their function is constantly needed in these microorganisms. So because of that, the function of these enzymes which are playing an important role in central metabolic pathways, their functions, they are referred to as housekeeping functions. And those genes which are participating in these housekeeping functions, these genes, they are referred as housekeeping genes. And the process through which these genes, they are continuously expressed, this process is known as a constitutive process, and the genes, they are also known as a constitutive genes. Means these genes, they are continuously expressed. There is no particular condition is needed for these genes to be expressed. They are constantly expressed in these bacterial cells. So because of that, they are called as a constitutive genes. Okay, so this is the one type of regulatory mechanism which is present in prokaryotes. Then another mechanism is also there. Now many genes are there which are expressed only when needed, and their gene products level, it may rise, it may fall according to the conditions, according to the molecular signals, and this is known as a regulated gene expression. Means if hard condition is there, such you can say that there is a lack of nutrient is there in that bacterial cell, in that bacterial environment, there is a lack of nutrient is there, and this bacteria is capable of producing endospores. In such conditions, then bacteria senses the condition that is there is a less nutrient is there. So according to these conditions, bacteria starts developing the resistant endospores. So at that time only the endospore production genes, they are expressed. So whenever the condition needed, at that time only these genes they are expressed, and this gene expression is known as a regulated gene expression. Now the gene products they may increase under particular molecular situations, means inducible situations. Some situations are there which induces these prokaryotes to express these genes, and such system is known as an inducible system. And the process of increasing the expression of their genes is known as induction. Okay, so if there is a signal is there that concentration of this protein is to be needed there, a signal comes to that process, and according to that signal the gene expression process initiate the increase of that particular component. So this is the inducible. But sometimes you can see the example, the expression of many of the genes encoding DNA repair enzymes. Now DNA repair enzymes, they are produced when there is a high level of DNA damage is there. This is the induced situation. According to this induced situation, in these prokaryotes, the DNA repair enzymes, they are expressed only in these conditions. When there is high level of DNA damage is there, this is the induced condition which is produced either because of the mutations caused because of various factors. So this condition induces the gene expression of those enzymes which are capable of repairing the DNA damage. So this is an inducible system where this particular gene expression is induced by particular incidents. Similarly, some gene products, they also decreases in concentration in response to the molecular signal. Okay, sometimes it may happen that because of some signals, the concentration of that enzyme, concentration of that protein, it get decreased, and such systems they are referred to as a repressible system, and the process is known as repression. A good example of this repressible system or a repression is the E. coli, where the tryptophan synthesis, this bacteria E. coli is capable of synthesizing the tryptophan amino acid by its own by following some biosynthetic pathways. And this tryptophan, when the concentration of this tryptophan is abundant in the bacterial environmental conditions, then the synthesis of tryptophan is totally stopped. When the concentration of tryptophan outside the environment, in that bacterial environment is low, at that time tryptophan synthesis biosynthetic pathway it started there. So this tryptophan itself acts as a inducer and as well as a repressor. When the concentration of tryptophan is low, tryptophan induces the biosynthetic pathway. When the concentration of tryptophan is very high, in such conditions, this tryptophan reduces the gene expression of tryptophan biosynthetic pathway. So these repressible and inducible systems, they are present, and as well as constitutive systems, they are present in prokaryotes in order to regulate their gene expression according to the environmental conditions. So in case of prokaryotes, gene regulations, they are carried out at different levels. As you know that in gene, which we have seen in earlier classes, that gene expression involves two important steps. First step is the transcription process through which the genetic message is transferred from DNA to a messenger RNA with the help of RNA polymerase enzyme. Secondly, the translation level, where the message from the mRNA is read by the ribosome, and according to that, the proteins, the amino acids, they are added in that protein molecule. So these two steps, they constitute the gene expression steps. Similarly, this gene regulation is also carried out at these two different levels, transcription level and translation level. So one by one we will say this. First, the transcription level, control mechanism. Now in bacteria, there are several mechanisms to control the gene expression at the transcription level. And this mechanism depends upon the gene product, whether that gene product is enzyme, whether that gene product is a particular protein which functions in a structures. According to that, the condition, according to these conditions, the regulation of gene is carried out at the transcription level. Now, this, you know that there in case of every organism, there are two types of pathways. They are existent, catabolic and anabolic pathways. These two pathways, they are constantly present in every living organisms. And so whatever the enzymes, they functions in these pathways, in these catabolic and anabolic pathways, they are regulated. For example, in multi-step catabolic system, the availability of molecule to be degraded commonly determines whether the enzyme in the pathway will be synthesized. We can simply take the example of EMP pathway, which is started by the glucose. Glucose is the substrate for that pathway. If glucose is not available, then the enzyme which is needed for glucose to get converted into glucose 6-phosphate. That is the beginning of EMP pathway, where glucose is converted to glucose 6-phosphate by using a 1 ATP molecule by enzyme. That enzyme we call as a hexokinase enzyme. If the substrate glucose is not available, then the hexokinase enzyme will not be synthesized. Similarly, in anabolic pathway or biosynthetic pathway, the final product is often a regulatory molecule. As we have seen in the example, that is a tryptophan is synthesized by the E. coli by following a biosynthetic pathway. And these biosynthetic pathways, they are carried out by few enzymes. And these enzymes, they are regulated by this tryptophan molecule. When the concentration of that tryptophan is low, then the enzyme which is required for the synthesis of the tryptophan, they are started. So these are the two pathways which are regulated by the synthesis of the gene expression, the gene product, that is either by enzyme or by the proteins. Now similarly, as you know that in case of prokaryotes, there may be presence of a monocystronic mRNA or a polycystronic mRNA. If the monocystronic mRNA is there, produced, that the product of this monocystronic mRNA produced is the single polypeptide. The single polypeptide is also controls the expression of that gene. If the concentration of that protein is very, very high, then protein itself acts as a controller and reduces the gene expression of that particular protein. Again, the concentration comes down. It also begins the gene expression. So, this single polypeptide, which is produced from monocystronic mRNA, it can enhance or reduces the gene expression which is required for the production of that particular product. Now, the molecular mechanism for each of the regulatory way differs greatly and are of following two types. This gene regulation at the transcription level is carried out by two different ways. First, negative transcriptional control. Second, positive transcriptional control. These are the two mechanisms which are involved in the regulation of genes at the transcriptional level only. And the proteins which are involved in the regulation of these two mechanisms, that is negative transcription control and positive transcription control, these proteins which are involved in controlling these two pathways, two mechanisms, they are called as a regulatory proteins. And this action of these regulatory protein is most often responsible for induction and repression. If regulatory proteins means this regulatory protein senses the conditions and according to that it controls the regulation process. Means these are the regulatory proteins which are playing very important role in gene regulation at the transcription level. Now these regulatory proteins, they can exert negative as well as or positive control. Means these regulatory proteins can control the gene expression in a negative and a positive manner. Now these negative and positive regulations, they can, in some systems, they can control the gene regulation in two different ways, but in some systems, both the negative and positive regulation system, some systems, they can be regulated by positive regulation as well as negative regulation. But some systems are also there, which are regulated by only negative transcription control mechanism. Some systems are also there which are only regulated by positive transcriptional regulation mechanism. But some systems are also there which are regulated by both positively and negatively transcriptional mechanism. And in these conditions, they uses two regulators to respond to the two different conditions in the cells. Some systems where both negative regulation is also there and positive regulation is also there. In such conditions, two signal molecules or two regulators are there, and these two regulators means two types of proteins are there which functions in positive as well as negative regulation of that particular system. In a catabolic system, which may be regulated by positively or negatively. Same example is that if glucose is available, the enzyme is produced, hexokinase enzyme is produced, and this hexokinase enzyme then converts glucose to glucose 6-phosphate. If glucose level get decreased, then this expression of this hexokinase enzyme is stopped. Similarly, in anabolic pathways, the final product usually regulates negatively its own synthesis. Example is that of tryptophan amino acid, which is synthesized by biosynthetic pathways or anabolic pathways, and this tryptophan regulates its gene expression required for the enzymes which are involved in this pathway. This tryptophan regulates this tryptophan biosynthetic pathway in a negative manner. Sometimes absence of the product also increases its synthesis. Means you can know that if tryptophan level get decreased, then tryptophan itself act as a inducer and enhances the gene expression which is required for the product, which is required for the enzymes of that biosynthetic pathway, of tryptophan biosynthetic pathway. So these two negative and positive regulation systems, both the systems, they are present in a mechanism. In anabolic pathway, this negative regulation is also there, as well as positive regulation is also there. In catabolic pathways, there is also this catabolic systems. It is also may be regulated by negative mechanism or positive mechanism. Now, the negative transcriptional control occurs when a protein inhibits the initiation of transcription process. Now, you can simply say that this negative transcription control mechanism, how it performs, how it controls the gene regulation. The regulatory protein which is involved in negative transcriptional control mechanism, that protein we call it as a repressor protein. And the function of this repressor protein is to inhibit the initiation of transcription process. If transcription is not initiated, the gene is not expressed. So this regulatory protein decides whether that gene is to be expressed or not. And that depends upon the particular condition which that bacterial cell faces. Now, in case of positive transcriptional control mechanism, it is also regulated by a protein, and that protein is known as actuator protein. And this function of this actuator protein is to promote the transcription initiation. Means negative transcription control mechanism, it inhibits the initiation of transcription, means directly inhibits the gene expression. Positive transcription control mechanism, where the actuator protein, it promotes the initiation of transcription process, means initiation of gene expression process. So this actuator initiates the transcription process, repressor inhibits the initiation of transcription process. So in case of negative transcription mechanism, this regulatory protein which we call a repressor protein, it functions by inhibiting the process of initiation of transcription. And in case of positive transcription control mechanism, the regulatory protein, that is the actuator protein, it promotes the gene expression by enhancing the binding of RNA polymerase enzyme to the binding sites on the DNA molecule. So these two functions in two different manners. In negative transcription control, repressor inhibits gene expression, while in case of positive transcriptional control mechanism, this actuator protein enhances the gene expression. Now these repressor and actuator proteins, they usually acts by binding DNA at specific sites. Why this repressor and activator proteins binds to DNA? Because as you know that whatever the information which is present on mRNA, it is taken from the DNA molecule. If this repressor and activator proteins, they get bound to the DNA molecule, it decides whether that gene is to be expressed or not. So, for repressor and the activator proteins, there are specific binding sites are present on the DNA molecule, on that gene. And according to the condition, this repressor and activator proteins, they get bound to that particular binding site on the DNA molecule, and it decides whether that gene is to be expressed or not. Here, repressor proteins binds a particular region on the DNA molecule that called as operator. The repressor proteins bind to a particular region on the DNA molecule of that gene, and that binding region for the repressor protein is the operator, which is usually overlaps or is downstream to the promoter. Means it is present nearer to the coding region, or it may be present after the promoter region. And when this repressor protein get bound to that binding site, that is the operator, when this repressor protein get bound to the operator, it inhibits the binding of RNA polymerase enzyme to the promoter, or it prevents its movement. So this repressor protein, when it get bound to the operator site of that gene, and after binding there, it blocks the binding of RNA polymerase enzyme to the promoter. As we have seen that in case of transcription, this RNA polymerase hollow enzyme, it only binds to the particular region on the gene, and that region we have called it as a promoter region. If promoter region is blocked by this repressor protein, then the RNA polymerase enzyme is not capable of binding there and initiate the transcription process. So this repressor protein functions by blocking the binding of RNA polymerase enzyme to the promoter. While the activator protein, which is involved in the positive transcriptional control mechanism, it has a binding site. That binding site is known as the activator binding sites. And these activator binding sites, they are present upstream of the promoter, that is far away from the coding region. So for the activator proteins, which is involved in positive regulation, there is a binding site. That binding site is called as activator binding sites. And these activator binding sites, they are present far away from the coding region, far away from the promoter region. Means before the promoter, there is an activator binding site, and after the promoter, there is an operator, which is the binding site for the repressor protein. And now the binding of this activator to the regulatory region, it promotes the binding of RNA polymerase enzyme to the promoter. The function of this activator protein is to bind to the activator binding sites. After it gets bound to that activator binding site on the DNA molecule, it promotes the binding of RNA polymerase enzyme to the promoter. Means beginning of the initiation process of transcription. So these two proteins, their functions opposite to one another. In case of negative control, the repressor proteins binds to the operator and avoids the binding of RNA polymerase to the promoter. While this activator protein, when it gets bound to the activator binding site on the DNA molecule, it promotes the binding of RNA polymerase enzyme to the promoter of that gene. Means directly initiate the gene expression process, that is the transcription. Now these repressor proteins and the activator proteins, they are present in two different forms, active and inactive form. And if the transcription is initiation, that depends when that repressor and activator proteins they are present either in active or inactive form, that decides whether the transcription is to be started or not. The activity of regulatory protein is modified by some effector molecule. Suppose some molecule is there, or we take an example of lactoperon, which is needed for the utilization of lactose sugar, which is present in the surrounding environment. And that lactose sugar, presence of this lactose sugar decides this expression of lactose operon, means those genes which are producing the some enzymes which are required for the utilization of lactose, that is controlled by the presence of lactose in the environment. So here the lactose molecule, it acts as a effector molecule, and when this activity of this regulatory protein is also controlled, it is also modified if some effector molecule bounds to the regulatory protein. For example, a repressor, some effector molecule comes and binds to the repressor. That repressor gets released from the operator, so operator is free. Then the RNA polymerase holoenzyme can easily bind to the promoter and initiate the gene expression. So this effector molecules decides whether that regulatory protein, either repressor or the activator proteins, it may be active or inactive. When these proteins they are present in inactive form, they are not performing their functions, means gene expression is there. When these proteins they are present, when repressor protein is in active form, gene expression process is totally stopped. When the activator protein is present in activator form, gene expression is taking place. So this effector molecule decides which condition is there. Now there are four basic ways in which the interaction of the effector and a regulatory protein can affect the transcription. Four different ways of gene regulations are there at the transcription level by these two mechanisms, negative and positive transcriptional mechanism. By these two mechanisms, gene expression at the transcription level, they are controlled. And as you know that there are three systems are there in bacteria. Inducible system is there, repressible system is there, and constitutive system is there. Now this inducible and as you know that the constitutive system, it is constantly there. The expression of these constitutive genes, they are compulsorily needed there because of the central metabolic pathways. These enzymes, they are constantly expressed. But in case of inducible and the repressible system, these genes, inducible genes or the repressible genes, they are expressed only when suitable condition is there. So depending upon that, there are four different ways are there through which the gene expression at the transcription level is controlled. First way, for a negatively controlled inducible genes, for example, those genes which are encoding enzymes needed for a catabolism of a sugar molecule. Example is that of glucose. Glucose is a sugar which is involved in catabolism. Or another sugars are also there. Arabinose is there, ribose is there, fructose sugar is there. Now in such condition, the repressor protein is active and prevents the transcription when the substrate of the pathway is not available. For example, every central metabolic pathway in a bacterial cell, it begins from the glucose. But other sugars are also there. If glucose is not available, then bacteria has to use the other sugars. In such conditions, if fructose sugar is there in the environment, no other sugar source is available, then bacteria has to use that fructose sugar. So, for utilization of this fructose sugar, whatever the enzymes they are needed, they are not expressed until fructose sugar is present in the environment. Okay, so this is the inducible system. Fructose is present, no other energy, no other sugar source is there. At that time, fructose acts as an inducer, and according to that, the gene expression takes place for the utilization of this fructose sugar. Whatever the enzymes which are required for the utilization of this fructose sugars, they are expressed when fructose is present in the surroundings. If no fructose is there, at that time, the gene expression which is required for the fructose utilization is stopped. Means the substrate decides whether that regulatory protein is active or inactive. Now this repressor protein, it is active, it binds to the operator and blocks the binding of RNA polymerase enzyme to the promoter. When the fructose is not available in the medium, so there is no requirement for the enzymes which are required for the utilization of the fructose sugar. So it blocks them. This is the negatively controlled inducible system, inducible gene. Similarly, negatively controlled repressible genes, they are also controlled. Those genes which are encoding enzymes needed for the synthesis of a particular amino acid. Take an example of tryptophan. The repressor protein is initially synthesized present in an inactive form. Okay, in case of negatively controlled repressible genes, the regulatory protein, that is a repressor protein, it is present, it is synthesized and it is present in inactive form. And when it is present in inactive form, that repressor protein is known as aporepressor. Okay, this protein which is present in inactive form, it is known as aporepressor. And this aporepressor, which is the inactive form of repressor protein, it gets activated after binding to the co-repressor. For example, we take an example of the tryptophan biosynthetic pathway. If tryptophan level is high, here tryptophan acts as a co-repressor and get bound to the aporepressor, and then it forms the repressor protein, active repressor protein. And this active repressor protein get bound to the operator and blocks the RNA polymerase binding to the gene. So these are the two different ways through which these two different systems, they have been controlled. The repressible system and the inducible system. You can see here, this is the negative control of inducible gene, where the repressor protein get bound to the operator, means no substrate is there. At that time, the repressor protein, it is bound to the operator, so there is no binding of RNA polymerase enzyme to the promoter, no transcription. If the substrate is available, that substrate act as an inducer, get bound to the repressor protein and get released from the operator side. So the promoter and the operator sites, they are free. In such conditions, the RNA polymerase enzyme get bound to that promoter region and begins the transcription process. So this is the negative control of inducible genes. Means here the substrate decides, means effector molecule decides whether that repressor protein remain there at the binding site or not. If repressor protein remain at the binding site, then the transcription process is not there. It's not occurring. If the substrate is available, if the lactose is present in the medium, here the lactose get bound to the repressor protein, then this repressor protein get free from the operator side, so the transcription process begins. Similarly, negative control of repressible gene, where the repressor protein it is present in inactive form, that is aporepressor, means in that medium, in that environment, there is a low amount of tryptophan is there. Okay, in equalized surrounding, there is a low amount of tryptophan is there. So the genes which are required for the production of enzymes which are involved in the tryptophan biosynthetic pathways, they are expressed. When the concentration of the tryptophan in the surrounding and inside the bacterial cell, it is present in high amount, then the tryptophan act as a co-repressor which binds to the aporepressor, so that repressor protein becomes active and it gets bound to the operator region. So there will be no binding of RNA polymerase enzyme to the promoter, no transcription process occurs. So in case of negative control of a gene, repressible gene, the repressor protein, it is present in inactive form. Only it is get activated when a particular substrate is there, and that substrate get bound to that aporepressor, so that it becomes active repressor protein and get bound to the operator site. So this is a negative regulation of these two inducible and repressible genes. Similarly, here the activator proteins, it regulates the gene expression of induced gene in an activated manner. Means it here the inducer comes, binds to the activator protein, and that activator protein gets bound to the activator binding site. Similarly, this activator protein controls or regulates the inducible as well as repressible genes. You can see here in this figure. So these are the promoter site. Far away from the promoter site, there is a presence of activator binding site, which is a specific binding site for the activator protein. When this activator protein it is present free, means it is present in inactive form. And when it is present in inactive form, there will be no transcription occur. But an inducer comes, binds to the activator proteins, then it gets converted into an active form and binds to the activator binding sites. So after binding of this activator protein at the activator binding site, it promotes the binding of RNA polymerase to the promoter and begins the transcription process in an inducible gene. But in case of repressible genes, this activator protein, it is already present in active form. Okay, it is already present in a active form and get bound to the activator binding site and it promotes the transcription process. But an inhibitor comes and bounds to the activator protein. It gets released from the activator binding site. No transcription occurs. So these are the gene regulation at the transcription level. These bacteria, they control their gene regulation at the transcription level by using these two processes, positive regulation and the negative regulation system. And by this system, they are capable of controlling the gene expression in case of inducible genes and the repressible genes. So that's it for